Hello, I am Dr. Aizaz from MedicoVisual.com, back with the second part of insulin resistance pathogenesis. So we have already discussed that the obesity, it is linked with insulin resistance and type 2 diabetes mellitus. Now the question is that how obesity causes insulin resistance. To understand this, let's go to first the normal physiology and normal biochemistry, a little bit into normal physiology and biochemistry of lipids and fats. So, the fats, they are stored in our body in specialized fat storing cells, and these cells are called adipose cells or adipocytes. So fat cells or adipocytes. Now these fat cells, they are not just a ball of fats, they are cells actually, and within these cells, of course, there will be nucleus and other organelles, and there is cytoplasm as well, and along with that there is a relatively large lipid droplet that stores the lipid inside the cell. Now here is the problem. The lipids are hydrophobic in nature, and we have different components of the cell that have some hydrophobic parts. For example, let's suppose here there is a protein, and this protein, it is made up of different amino acids. So some of these amino acids, they may be, let's say, hydrophobic, and let's say here there is a hydrophobic amino acid, and here there is another hydrophobic amino acid. So they were interacting with each other, and they were forming a particular three-dimensional structure. But if a substance with relatively large hydrophobic nature, which is much more hydrophobic as compared to let's say this amino acid, so rather than this amino acid interacting with this, it might interact with this lipid, with these lipids, so the structure of proteins might disrupt. So what we can say is that we will discuss these details later as well, but here the idea is that the lipids, they can disrupt the cellular components of the cells. So that is one of the cause of lipotoxicity or lipotoxicity. To prevent this problem, how can we safely store the lipids inside the cell? The mechanism is that they are compartmentalized. They are stored in special compartments. You might be having some gas cylinders in your house, right? But this gas might be dangerous if it is left to leak. So what you do is that you store that toxic things, that toxic chemicals and gases inside specialized containers, and these containers, they do not allow the leakage of those toxic substances. In the same way, these lipids, they are not as toxic as those toxic chemicals I am talking about, but still they can be damaging for the cell. So they are stored inside the lipid droplets, and these lipid droplets, they are surrounded by phospholipids. The phospholipids are very interesting molecules. They have this let me show you. So they have this hydrophobic part. Here is this hydrophobic part, and they have this hydrophilic part as well, which is polar head. So they have hydrophobic part, this one, and they have hydrophilic part. So both parts are present in a single molecule. So because of this reason, at one end, from the hydrophobic end, they are interacting with lipids, and from the from the polar head, it is interacting with the aqueous environment of cytosol. So they are like dual-faced hypocrites. They are interacting with both of these structures. So this is, let's say, phospholipid, and it is interacting with, on one end it is interacting with non-polar molecules, and on the other end it is interacting with polar molecules. So this was a meme just for your fun purposes, so that you can remember it forever. So let's move to back to the lecture. So we have this lipid droplet which is surrounded by single layer of phospholipid. Remember our cell membrane, it is lipid bilayer, but this is not bilayer, this is unilayer of the phospholipid. Of course, there is, like our cell membrane, there is cholesterol as well in between this, which I am not shown. And now something interesting here. These adipose cells or adipocytes, they have special molecular cutters. They have special enzymes that can cut the fats that is stored inside the lipid droplets. And in what form these fats are stored in lipid droplets? Mainly these are triacylglycerols. We will discuss what is triacylglycerol later. Of course, there are some other fats, some other minor types of fats as well, like diacylglycerols and cholesterol, esters, cholesterol, etc. But mainly that is triacylglycerol. Right? So these cutters, they can access this lipid droplet, and they can start cutting the triacylglycerol, and they can ruin all that lipid stores that were present inside the cell. So this process should be controlled, and as it turns out, it actually is tightly controlled. These cutters, they are not allowed to unduly access the components of this lipid droplet. So how this happen? Because this lipid, it is protected by special guards. So it is guarded by these guards that surround this lipid droplet, and they do not allow the undue access of these lipases. By the way, these cutters, because they cut the lipids, they are called lipases. So undue access of these lipases to the fats of this lipid droplet is prevented by these special guards. And what are the name of these guards? Basically, these are proteins, and they are called perilipin 1 or perilipin A. There are some other types of perilipin as well, and they are a component of different parts of different cells of the body. Even the perilipin 2 may be in adipose tissue as well, but their function is not very well understood at the time of recording this lecture. So I will refrain from discussing those details in this lecture. And by the way, remember that this perilipin, it is specialized mainly for adipocytes. At some other places, this perilipin 1 is also present. For example, I think in adrenal cortex perilipin is present, but mainly it is the part of adipocyte. And here the function of this perilipin 1 is that it guards the lipid droplet. Now, if this lipid droplet is guarded all the time, then how can we get the free fatty acid? How can we use these stores? Of course, these stores are not there to remain there as a store forever. We must have access to these lipids when needed. So how these free fatty acids are released into bloodstream at the time of need? So let's discuss that. You must be wondering that why I am discussing these things. Because if I explain these things to you first, then it will be very easy to understand that how the obesity is linked with insulin resistance. So remember, my friends, the basic concepts, the basic sciences, they are very crucial and very important to understand the clinical medical sciences. So actually, when these free fatty acids are needed in the blood inside the body to be utilized for oxidation, for example, in stress conditions, in stress situations, certain hormones are released into blood. For example, mainly catecholamines are released. Of course, there are certain other hormones as well, but mainly let's suppose catecholamines are released. Now, catecholamines, of course, again, they are peptide hormones. They cannot go inside, so they will bind to their G protein couple receptors, and through multi-step pathway, they will wake up, they will activate their friends inside the cell, and they are called, what is the name of their friend? That is called protein kinase A. So what protein kinase A will do? Basically, this catecholamines, they are ordering the protein kinase A that, hey dear friend, I need free fatty acids inside the blood. So do something with these guards. These guards are guarding this lipid droplet, and they are not allowing these cutters to remove the free fatty acid from triacylglycerol. So these protein kinase A, they are quite cunning. What they will do is that they know how to deal with these guards. So they will bribe these guards with phosphates. So some of these guards, they will be given money, not money, they will be given phosphates. I mean, they will be phosphorylated, and of course, the number of guards that will be phosphorylated depends upon the number of protein kinase A that are activated inside the cell, and that depends upon the amount of catecholamines that are stimulating. Of course, there will be a number of cell receptors, and if more cell receptors are stimulated, they will activate more protein kinase A, and more guards will be phosphorylated. Not guards, perilipins. So more perilipins will be phosphorylated. So as soon as they get bribed, they get phosphorylated, they will leave this path and they will move aside. Not only that, but they will send a special key to this molecular cutter, and it will activate this cutter. This key is basically the cofactor of this molecular cutter, and if you want to know the name of this key, name of this cofactor, it is CGI58. So by the way, this information, you might be interested in that, but I don't think you need to remember it at this point, at least for the insulin resistance. So anyhow, so what they have done, these guards, they have paved the way for these lipases to come and attach with lipid droplet, and they have also activated this lipid droplet by releasing special cofactor called CGI158. So as they become activated, they will be translocated to lipid droplet. And as I have told you that this lipid droplet, it consists of mainly of lipids are stored mainly in the form of triacylglycerol. So what is triacylglycerol? It consists of this glycerol molecule, and with this glycerol molecule we have 3 fatty acid chains connected. So glycerol backbone with 1, 2 and 3 fatty acid chains connected to this. So this is triacylglycerol. Now what this lipase will do is that from this, it will act upon this triacylglycerol of this lipid droplet, and it will remove 1 fatty acid either from this position 1 or from position 3. So here you can see that it has removed this 1 free fatty acid, in this case from position 1. It can also remove from position 3 as well, right? So now we have diacyl, because 2 fatty acid chains, diacylglycerol. Now we have diacylglycerol and 1 fatty acid. Because this lipase, it is acting upon triacylglycerol, so it is called adipocyte triacylglycerol lipase. What it is called? Adipocyte. So here is A for adipocyte, T for tri, G for glycerol. It is also called, by the way, triglyceride. So adipocyte triglyceride lipase, right? Then now we have this diacylglycerol, and with this diacylglycerol we still have 2 fatty acid chains connected. So we need to fetch even these 2 free fatty acids as well. We need to free these 2 fatty acids from these diacylglycerol. So we have another lipase, and this lipase, these protein kinase A, they also bribe this lipase by phosphorylating it, and then this lipase, it will first go to actually to the lipid droplet, but for the sake of simplicity I have not shown it going to the lipid droplet, because these diacylglycerol, they will still be the part of lipid droplet. They will not come into cytosol, but it will become very you can say congested diagram if I drew it here. So I have drawn it here. So this diacylglycerol, again from 3 position or from first position, this free fatty acid will be removed. So we have now another free fatty acids and monoacylglycerol molecule. And finally, because this is diacylglycerol, this is acting on diacylglycerol, this is called diacylglycerol lipase or diglyceride lipase. Now here's something interesting. Initially, when scientists were studying these lipases, they first found this lipase, this diglyceride lipase, and what they found is that this diacylglycerol, it brilliantly respond to hormones. For example, it will very nicely respond to presence of catecholamines, even to presence of glucagon, cortisol, and many different hormones. So they named this lipase, and remember at that time this was the only lipase that was discovered inside the adipocyte. So they called this lipase as hormone sensitive lipase or HSL. But remember, almost every lipase inside the adipocyte is hormone responsive, it is hormone sensitive, but traditionally diglyceride lipase is the one that is called hormone sensitive lipase. So if someone asks you that what hormone sensitive lipase does, you should say that it acts on diacylglycerol of the lipid droplet and converts it into monoacylglycerol and free fatty acid. But still there is some evidence that this hormone sensitive lipase, this diglyceride lipase, it can also act directly upon triacylglycerol as well. Just like this ATGL, it can directly act upon triacylglycerol as well. So by the way, this was just side information. So let's move forward. Now this monoacylglycerol, it still has one fatty acid that we need to free. We need to free up, and again there is monoacylglycerol lipase that will release the glycerol and another free fatty acid. So what is the end result? In the end we have 1, 2 and 3 fatty acids and a glycerol molecule, and all these 4 molecules, they will be released into the bloodstream and they will raise the free fatty acids level in the blood. Now remember, this free fatty acid, it is a misnomer. Actually the free fatty acids, they shouldn't be allowed to freely move into the bloodstream, because they might clump together because of their hydrophobic nature. So basically we have special transporters inside our body, the albumin. So albumins are special transport vehicles. What they will do is that they will pick up this free fatty acid. So free fatty acid will be sitting on this vehicle, and then they will transport this free fatty acids to different parts of the body. And remember, one albumin molecule can bind up to 10 free fatty acid molecules, right? So that's how normally the free fatty acids are released into blood. That is totally normal phenomena. Now here is the thing, after puberty the number of adipocyte, it does not increase significantly. Of course there is some increase in number of adipocyte after the puberty as well, but that is not very significant. So for the sake of our discussion, let's assume that the number of adipocyte will remain constant throughout the adulthood. Of course that is not entirely true. So let's focus on one adipocyte. Now if this person is eating too much calories, all these extra calories will be stored in his or her adipocytes in the form of triacylglycerol or triglycerides. So the number of this lipid droplet, not number, I mean size and volume of this lipid droplet, it will continue to gradually increase. So as this happens, if the number of these guards, these perilipins, it remains same, do you think that same number of perilipins will be able to guard this larger larger lipid droplet? The answer is no. So to compensate this, the body will synthesize more perilipin, but even when the lipid droplet size is very large, its surface area is very large, even the increased synthesis of these perilipin, they will not be enough to guard this perilipin properly and adequately, because the synthesis, it can occur up to a certain limit. So what will be the end results? There will be some areas at which these lipases, they will come and they can interact with these triglyceride, and even without hormonal stimulation, perhaps because of increased substrate concentration, they will act upon these triacylglycerols and they will convert them first into diacylglycerol and then monoacylglycerol, and of course the end result will be that there will be increased release of free fatty acid and glycerol molecule even without the hormonal stimulation, and these free fatty acids, they will be raised in the bloodstream. We do not know the precise reason that why it happens, why and how exactly these lipases, they get activated without hormonal stimulation. But one of the hypotheses is that this is because of increased substrate concentration. And here there is some importance of genetics as well. For example, in a person who is genetically more prone to development of insulin resistance, and there might be some abnormality, there might be some variation in perilipin, so as that this perilipin, it might not be very efficient in guarding this lipid droplet. So even a little increase in the size of this lipid droplet, it makes the person more prone to increase lipolysis. And of course there is the role of perilipin, there is the role of these lipases, and genetic variation in any of the components of these substances, these molecules that are involved, they may make a person more prone or less prone to development of insulin resistance. So that is why genetics are very important. They play a crucial but not the sole role in development of insulin resistance. So what we can say is that in this case, even though the number of perilipin, it might increase, but the number of perilipin per unit surface area is not adequate, it is less than normal. So let's suppose in this area, let's say we need, 5 perilipin molecule were needed, but we only have 3 perilipin molecule, 1, 2 and 1, 2, so rather than 5 we have 3 and 2 are missing. So because there is relative deficiency, this is not absolute, this is relative deficiency of perilipin, this might lead to a problem, and that is called increased basal rate of lipolysis. So what is the problem here? So there is decreased perilipin per unit surface area of the lipid droplet, which led to improved excess of resting adipocyte, triglyceride lipases and other lipases to the triacylglycerol molecule of the lipid droplet, which led to increased basal free fatty acid level in the blood, or we can say that there is increased rate of basal lipolysis in the adipocyte. So that is a problem. So end result is that we have increased free fatty acids in the blood. Now the next question should be that why increased free fatty acid is the culprit behind development of insulin resistance. So let's see how free fatty acids causes increased free fatty acids cause insulin resistance. So let's suppose we have lots of free fatty acids in our bloodstream in the extracellular fluid, and here is a cell. So as free fatty acids are raised outside the cell, there will be increased entry of free fatty acids inside the cell by its transporters. So we have fatty acid transporters here which will transport through facilitated diffusion. They will take the free fatty acid inside the cell. So naturally, if there is increased free fatty acids in the blood, more free fatty acid will enter inside the cell, because it is diffusion. Even though it is facilitated diffusion, but it is still diffusion process. And diffusion, by definition, diffusion is transport of substances according to the concentration gradient. If the concentration gradient is higher, there will be increased movement of free fatty acid from outside the cell to inside the cell. So free fatty acids, what they will do is that they will move into the mitochondria. Now inside the mitochondria, they will get oxidized by the process of beta oxidation. We will discuss the detail of beta oxidation sometime later, but here what the free fatty acids will do is that they will, as there is more free fatty acid, they will increase the rate of beta oxidation of free fatty acids because of two reasons. One reason is very simple. There is more substrate, that is more free fatty acids are there for being oxidized. As the raw materials are more, so more will be the product, right? The other reason is that there is some evidence that free fatty acids, if there is prolonged free fatty acids inside the blood and inside the cell, they will activate and they will upregulate the synthesis of enzymes that are required in beta oxidation, that are required for beta oxidation. So during beta oxidation of free fatty acids, a lots of acetyl-CoA molecules will be released. How that will be released, that is not under the scope of this lecture. We might discuss that later. Along with that acetyl-CoA molecule, lot of acetyl-CoA molecules will be released, and also some high energy, high electron reduction products called NADH and FADH2. They are also formed through multi-step process, of course. Now these NADH and FADH2, they will run through electron transport chain. They are basically a complex of enzymes and proteins, and these FADH2 and NADH2, they will run through these complexes of proteins, and as a result of this, a lots of ATP molecules will be generated. So what we have got as a result of beta oxidation of fatty acids? A lots of acetyl-CoA, a lot of NADH and FADH2, and a lots of energy currency, the ATP. Now inside the mitochondria, we also have oxaloacetate, and this acetyl-CoA, it will combine with oxaloacetate to form citrate, and this cycle might continue to run, and it will continue to form more and more ATP, and more NADH and FADH2 will be formed, and these molecules, they might go to this electron transport chain to generate more energy currency in the form of ATP. Now if there is too much energy, too much ATP, too much NADH inside the cell, this cycle will stop at this point. Because as there is too much energy already inside the cell, we do not need to create more energy inside the cell. So naturally this cycle should be blocked here. It should not be allowed to continue. We already have too much energy, so let's put a break on this cycle. So that's what happens here. So as citrate will be blocked here, so citrate will come out of mitochondria through certain transporter into the cytosol. Of course, ATP will also come out and NADH will also come out. So all these components, they will be raised inside the cell, inside the cytosol as well as mitochondria of the cell. Now you see what happens. What insulin does is that it increases the uptake, utilization and storage of glucose. Insulin is released when we have too much glucose inside the blood, and we need to lower the glucose level inside the blood. So let's suppose this is a peripheral cell. This could be any cell, but classically let's suppose this is a muscle cell that we are talking about. So GLUT4 will be there, increased expression of GLUT4 on the cell surface will be there, which causes the entry of glucose from the extracellular fluid, that is blood, into the cytosol. And this glucose, it will also be oxidized by the process of glycolysis into pyruvate, and then pyruvate will ultimately form the acetyl CoA. Of course, as the glucose is being utilized, and ultimately when there is not much need of energy, this glucose will also be converted into the storage form, into its storage form called glycogen, and that process is also stimulated by insulin. Now you see, now let's focus here. What insulin is doing? Insulin is causing increase in pyruvate, and pyruvate leads to increase in acetyl CoA, which leads to increase in citrate, and which also leads to increase in ATP, NADH, and ATP and NADH are also formed here through glycolysis as well. So you remember this thing. Now here is a problem. Free fatty acids were also creating all these substances. Glucose is also creating all these high energy substances. So do you think these two oxidation of these two fuels should go on side by side? I don't think so. This should happen. This is just like you are running a car, and in this car at the same time you are using petrol as a fuel, I mean gasoline as a fuel, and at the same time you are also using diesel as a fuel. So either you should use petrol or you should use diesel, not both at the same time. So here the body also does this. So these substances, what they will do is that they will block the utilization, they will block the glycolysis. These substances, they will block the glycolysis. They will say that, hey, free fatty acids have already created the high energy components for us. Why the heck you glucose are giving us that same things again and again? We cannot handle that much energy, that much high energy substances inside our body, inside our cells. So they will block this. So the end result is that utilization and oxidation of free fatty acids, that will block the glucose. And you know that this process is also true vice versa, that if there is increased utilization of glucose, it will inhibit the increased utilization and uptake of free fatty acids. But what is the mechanism? I will not go into detail, but just briefly let's talk about. You know, this process glycolysis, certain enzymes are involved here. For example, pyruvate dehydrogenase is here, then pyruvate dehydrogenase complex is here, then pyruvate kinase is here, then phosphofructokinase 1 is here, and hexokinase 1 is here. So several different enzymes are there that are involved in the process of glycolysis. depending upon the enzyme, that it depends upon enzyme that it is blocked. Certain enzymes are blocked by, let's say, citrate, certain are blocked by ATP, or even more than one components might block an enzyme. So what these components will do is that, let's suppose this is an enzyme. So here this, let's say, citrate is there. Citrate will come and it will block this, it will block this enzyme. So allosterically they will inhibit this enzyme. So due to allosteric inhibition of this enzyme, the activity of this enzyme is drastically reduced, which leads to decreased utilization of glucose, decreased oxidation of glucose in the presence of increased free fatty acid oxidation inside the cell. Now you see the situation here. So that is like a tug of war between free fatty acids and insulin. On one end, insulin is trying to force the cell to use glucose, but on the other end, increased free fatty acids, they are trying to force the cell to use the free fatty acids. Free fatty acids, they are resisting the actions of insulin. So this is one of the reason of insulin resistance. Now here, let me tell you something interesting, that not only the utilization but also the uptake of glucose will be reduced. Let's see how. So as the pyruvate accumulates here, similarly the other components of other metabolites of glycolysis, they will also accumulate. Ultimately, even the glucose will accumulate inside the cell, right? So as the glucose will accumulate inside the cell, so there will be decreased concentration gradient between the cell and outside the cell. So there will be decreased glucose concentration between extracellular and intracellular environment. So naturally there will be decreased uptake of glucose by the cells. So what is the end result? Let me remove these things. So end result is that we can say that glucose and free fatty acids, they compete with each other for uptake and utilization, for uptake and oxidation. They compete with each other. They can also inhibit the other process. So if glucose oxidation is going on, that will inhibit the free fatty acid oxidation, and if free fatty acid oxidation is going on inside the cell, that might inhibit the glucose oxidation, and this is called Randle Hypothesis, on the name of a famous biochemist and an esteemed medical researcher. His name is Dr. Philip Randle. I hope I am pronouncing his name right. I'm not good at pronunciation. So in his honor, we call this as Randle Hypothesis. There is also some evidence, by the way, that when there is increased free fatty acid inside the cell, they might directly inhibit the activity of GLUT4. And there is some evidence that they might directly decrease the translocation of GLUT4 on the cell surface. So through number of ways, increased free fatty acids, they resist the action of insulin. So this is one hypothesis. In next part of this lecture series, we will discuss another hypothesis, that how the free fatty acids and how obesity is linked to the insulin resistance. I hope you learned something from this lecture. If so, please like this video and please subscribe to my channel, and please do share this video with your friends and colleagues. Thank you so much for watching this video. I will see you in the next video. Here is Dr. Aizaz signing off. Bye bye.